Okay. So our first objective here is to describe the various properties of graded potentials, including the direction of change in a potential, the magnitude of change. We're also going to think about temporal and spatial summation. These are different mechanisms for adding or summing graded potentials. And lastly, how graded potentials in neurons can eventually lead up to or trigger an action potential. Okay, so we first need to think about a smaller potential, which is the graded potential. And so there are two main types of potentials that are essentially leading up to the bigger action potential. So graded potentials are going to be small. They're going to be short in terms of their distance, right? The distance that they communicate over, versus action potentials, which are larger, more substantial changes in the membrane potential. Action potentials are also going to communicate over long distances. And when we say distance here, we're meaning on the neuron. So the graded potentials will be initiated at the soma, which is the most proximal area of the neuron. This is where they start. And then they're not going to travel as far down the neuron as action potentials will. They're actually going to dissipate or lose their strength as they move away from the source. So that's one of the major differences between the graded potential and the action potential. However, an action potential is really triggered by a graded potential that is strong enough to get to threshold. So you can see that this number is marked here, negative 55. Basically, this is the threshold value that determines whether or not we trigger an action potential. So in the first panel here, we see some small changes in the membrane potential. These can be positive changes, which is going up, right? We're going from negative 70 and moving in a positive direction, or they can be negative changes, which are going below negative 70, right? So more negative. Regardless, those can be added. We're going to talk about summation. And if they're strong enough or they occur close enough in time and they get us to a negative 55 millivolt, right? They're enough of a change to go from negative 70 to negative 55. From there, we're going to trigger a full action potential, and that must be completed before the membrane gets back to rest at negative 70. So this is a more meaningful signal. The action potential, because it can communicate over long distances, and because it is a larger change in the membrane potential, this can communicate from neurons to muscle and are essentially a major route of signaling between excitable cells. So let's kind of think about where on our neuron we have these action potentials versus graded potentials, and also where we have different channels along the neuron. So let's describe and differentiate these different potentials. Let's begin by thinking about the structure of our neuron. So here's the proximal portion of the neuron. Here is the midpiece or the axon, and then the terminal. Follows nucleus. So this is the soma or cell body. This is where we have the cytoplasm, the organelles, the nucleus, etc. We also have dendrites, which are projections from the soma, right? So this would be an example of a dendrite. Okay. And we also want to think about the direction of signaling. So the dendrites, as well as the soma, are picking up signals from another neuron. So I'm going to draw the end terminal of another neuron in adjacent or in proximity to the soma so that we can see that the flow of direction, a signal is being received from one neuron at the soma, and then that is being projected to the axon terminal here, and likewise at a different synaptic junction. So the area in between one neuron and another where they're sharing signals, this region is called a synapse. Okay. Let's think about some other areas on this. Actually, let's kind of describe this here as well. So in relation to one neuron signaling another, we can call this one the presynaptic neuron. Right? Because it's before the synapse. And we can refer to this one as the postsynaptic neuron. Okay. Now let's begin to think about the other parts on the neuron. So we have the axon hillock. This is essentially the neck of the neuron, the axon hillock, right? Otherwise known as the neck or narrowing from the cell body to the axon. Okay. This is called the axon itself. And then this is the axon terminal. Right. Now let's think about the channels and their different locations along the neuron. So where we have the soma and the cell body, we have a lot of ligand-gated channels, right? Ligand-gated channels. So I'm going to put that here where we have the soma or cell body, we have ligand-gated channels. And this kind of makes sense, right? So the channels that are on the proximal portion of the neuron, something needs to bind to them for them to open up. And that's what happens at the synapse. We're going to talk about this in the next unit, but essentially neurotransmitter or chemical compounds are released at this synaptic junction and can bind to these ligand-gated channels. And that is how the signal goes from the presynaptic neuron to the postsynaptic neuron by way of opening up ligand-gated channels, and then the rush of ions is what creates a graded potential on the proximal part of the neuron. Let's look at the axon hillock. Here there's going to be an abundance of voltage-gated channels. Voltage-gated channels. And these are channels, they're also gated, but they're going to be open when the membrane potential gets to a certain value. This is where that -55 number comes into play. So when we have enough graded potentials through our ligand-gated channels to get to -55, that's the voltage where other channels on the axon hillock are going to open up because they're voltage gated. And that is where the signal can now move from the axon hillock down the rest of the axon. So these are going to be responsible for starting the action potential, from taking the signals that are formed by graded potentials and actually making a larger membrane potential change that can travel the distance of the axon down to the axon terminal. So these will be graded, voltage gated channels. If we think about the axon terminal, the type of channels here are called calcium channels. And we'll look later on at how these behave, but they're kind of concentrated down here at the axon terminal. And then I want to kind of make a note that throughout the entirety of the neuron, let's just put it over here. Throughout the entirety of the neuron, there are leak channels. So the channels that we discussed previously when we talked about the differing permeability, right, which are basically leaking sodium or potassium, those are found throughout the entirety of the neuron as they are important in establishing the resting membrane potential. So we don't want to forget about the leak channels. Okay. Likewise, we also do not want to forget the sodium-potassium pumps. So those are also present along our nerve. Now let's think about the difference between graded potential and action potential and just describe that here. Excuse me. So this is kind of the cutoff zone, right? This is the proximal part of the neuron, right? So proximal to the axon hillock, graded potentials are initiated. Which are smaller membrane potential changes. And at the axon hillock, we're going to assess those smaller graded potentials, and they can be coming in from different presynaptic neurons. So although we only drew one here, there can be many other presynaptic neurons that are sending their own individual signals, right? And the axon hillock is where all of that is sort of assessed. So we're going to calculate what are the different amplitudes of graded potentials coming in at these various synapses on the soma, and we're going to determine, is it enough to get us to -55? And if so, then we're going to initiate an action potential from the axon hillock onward. So the axon hillock onward is where we have action potentials. Okay, which are larger, which is a larger membrane potential change. And we're also going to say that it's for a longer distance. It's going to travel for a longer distance. Right? So this is the direction that our action potential will move until it gets to the axon terminal, and it's going to trigger the release of chemicals, which is really what's happening at the proximal end as well. So this is how neurons communicate. This is how they speak to each other by way of chemical and electrical signals that help one neuron to be able to signal the other neuron in proximity. And I do want to make a note here of the -55 millivolts, which is the threshold. This is called the threshold value because if we get to this value at the axon hillock, we fire an action potential, and if we don't, those graded potentials will simply disappear or dissipate.